Somatic hypermutation does not generate diversity in T cell receptors. Somatic hypermutation increases the diversity of all three complementarity determining regions of both immunoglobulin chains. Somatic hypermutation does not occur in T cell receptor genes, so that variability of the CDR1 and CDR2 regions is limited to that of the germline V gene segments. All the diversity in T cell receptors is generated during rearrangement and is consequently focused on the CDR3 regions. Why T cell and B cell receptors differ in their abilities to undergo somatic hypermutation is not clear, but several explanations can be suggested on the basis of the functional differences between T and B cells. Because the central role of T cells is to stimulate both humoral and cellular immune responses, it is crucially important that T cells do not react with self-proteins. T cells that recognize self-antigens are rigorously purged during development, and the absence of somatic hypermutation helps to ensure that somatic mutants recognizing self-proteins do not arise later in the course of immune responses. This constraint does not apply with the same force to B cell receptors, as B cells usually require T cell help to secrete antibodies. A B cell whose receptor mutates to become self-reactive would, under normal circumstances, fail to make antibody for lack of self-reactive T cells to provide this help. A further argument is that T cells already interact with a self component, namely the MHC molecule that makes up the major part of the ligand for receptor, and thus might be unusually prone to developing self-recognition capability through somatic hypermutation. In this case, the converse argument can be made because T cell receptors must be able to recognize self-MHC molecules as part of their ligand. It is important to avoid somatic mutation that might result in the loss of recognition and the consequent loss of any ability to respond. However, the strongest argument for this difference between immunoglobulins and T cell receptors is a simple one, that somatic hypermutation is an adaptive specialization for B cells alone, because they must make very high affinity antibodies to capture toxin molecules in the extracellular fluids. And that somatic hypermutation does not generate diversity in T cell receptors. Have a great day.